So welcome everyone tonight to the Cbtn Community Information session. For those that don't know me, my name is Jenna Lilly, and I am the Cbtn Executive Director. I've had the honor of being part of Cbtn since 2012, and I thank you all for taking the time to join us tonight.
Thank you for presenting, Jenna. Appreciate it.
Sure, of course. So for today's topics, what we hope to do is give you a nice overview of Cbtn, present to you the different research model that the Cbtn is using through sharing of resources to help get us all closer to cures for these children, and open it up to discussion and questions. We are super excited to make sure that this is a very informative call for you all and efficient. So please feel free to break in with a question, put questions in the chat, but please speak up and participate. For those of you that don't want to be recorded, you can keep your video off and you can just place your questions in the chat. We will be sharing this recording on our website with folks that weren't able to attend. So we do really hope that this is an open discussion for you all. So I'm going to start real quick with what drives us, and I think it's what drives many of us that are on this call. As many of you know, pediatric brain tumors remain the leading cause of disease-related death in children. And, you know, there's never been a drug developed to treat childhood brain tumors. This statistic also has not changed. And so the Cbtn wanted to take on that challenge and create a new research model that would hopefully accelerate discoveries and new treatments for children. And the mission is a global one. We hope to help children all over the world. Pediatric brain tumors is not just the leading cause of disease-related death in the US, but in many, many countries around the world. But through our mission and vision, we also have a set of core values that's focused on open science, global inclusion, and pioneering spirit. So the new research model that Cbtn was proposing was one that was made up of collaboration and sharing. Due to the vast number of pediatric brain tumor types and the volume of pediatric brain tumor patients being seen at any single hospital, we were at like a very basic challenge with the research. There wasn't enough materials to be able to do the research needed across all of the pediatric brain tumor types. So what the Cbtn leadership proposed was, could we all come together? Could we all put our resources together in one place and share them in real time? And this could give researchers immediate access to the materials that they needed to perform research, but also increase the volume of research that could be done at one time. Typically with one laboratory, there's one research project that's going on, and it does take years within the basic laboratory wet bench, and then it does take years for analysis. And we wanted to try to take that model, turn it on our head, and see if we could accelerate through scale. Could we have all the pediatric brain tumor specimens, preclinical models, and data in one place, highly standardized, curated, and ready for any researcher, young researcher, older researcher, any researcher that came in and said, I would like to run a pediatric brain tumor project. And so we thought that this was going to be a challenge, but I'm happy to say that from the initial 4 institutions that started this new research model in 2011, we are now at 21 member hospital institutions across the globe. We have a hospital in Italy, we have hospitals in Australia, we have hospitals across the US, and we have participation in China. And there's the model for CBTN is to continue to grow and include institutions within this model. So it was fantastic to be able to see that expansion. When we started in 2011, we had about 120 subjects enrolled in the CBTN and about 500 biospecimens. And I'm happy to report to date, in 2020, we've enrolled over 3,600 participants across all brain tumor types. We collect the whole gamut of brain tumors to ensure that any researcher that has an interest in a specific brain tumor type can be served. These little cards that you're seeing on the screen here, we have launched a new website to provide in real time the number of participants that we have by a specific brain tumor histology, so researchers can then understand the resources available. We had some questions that were posed on our biospecimens and why was it important to collect tissue samples. And so these are some of the questions and responses that we wanted to provide you all. Tissue is what we need to understand the biology of the tumor. It's actually tissue from the tumor. So in that tumor, as you all know, cancer has just unprecedented growth. But what is the mechanism that's actually happening within that tumor? We actually need the biospecimen to be able to pull out the genomic and proteomic information from that tumor to see what switches on that isn't supposed to be on, what switches off, what's actually going within the specimen. And as I said earlier, there really wasn't a lot of pediatric specimens, and much of the progress of, let's say, adult cancers that we all have seen with new drugs and new trials and new treatments is due to the volume of biospecimens that was available for the adult conditions. So the more tissues that we collect and analyze, it provides a larger statistical cohort for us to be able to understand the mechanisms that are going on in the tissue. And as I said, there's over 100 different types of brain tumors. So being able to collect across the board, across all hospitals and institutions, can really provide us with that comprehensive resource that the researchers need. And along with the collection of those biospecimens, we follow the patient through their journey and collect key data points from their treatment and from their outcome and follow-up. And collecting the clinical data and samples at multiple times across this journey is really unique to CBTN. We've been able, with this information, to see what a tumor looks like genomically before it gets treatment with chemotherapy or radiation. And then if there's a progression or recurrence, what does the tumor look like after that treatment? How did cancer get around those treatment methodologies? What new mechanisms did it turn on, or what things did it turn off to be able to come back after that treatment? And we're finding the tumor that a patient is diagnosed with and the tumor that the patient perhaps loses their fight with is a very different tumor. And so those biospecimens and that clinical data, understanding what type of therapy they received, is key data points for the researchers to understand how the cancer is really fighting against us. CBTN also collects post-mortem brain tissue. This is our partnership.
A question for you, if
yeah,
you don't mind.
Sure. Go right ahead.
Those changes that you see, are they driven by the treatment itself?
So they're definitely, so they are, it's impacted by the treatment. So the treatment may be successful with the first mechanism that was identified for that type of cancer. So we may have, let's say, an NTRK pathway within a tumor. We found we have a drug, you can turn off that NTRK mechanism, but then the cancer finds a new way to continue and go around that mechanism. So it can it it's you'll see the damage that the therapy does, which is I think a part that you're asking, and you'll also see how the cancer can evade that treatment. The treatments themselves, as many of us know, are very harmful. You know, cancer therapies, if they're not precision, are really trying to kill the tumor and then bring back all the normal cells after we've treated with very harsh chemotherapies or radiation to try to bring all of the growth mechanism within a child almost to a slow. So you'll see within the recurrence or progression additional mutations and variations, yes, due to the therapies, and also potential mechanisms for the way that the cancer is evading the therapeutic approaches that we provided.
Thank you.
Sure. And for the post-mortem brain tumor collections, we partnered with Gift from a Child from the Swifty Foundation. This is a really remarkable program where we are able to collect post-mortem brain tissue. This program is very critical, allows us to get normal brain tissue. In addition to what I was talking about earlier was that initial recurrence and then the tumor that they died from, really being able to understand the mechanisms that are evolving in the tumor genesis throughout the patient's journey. So I'm happy to say that, you know, from that beginning in 2011, where we were about 500 specimens and 120 patients, we're now at over 40,000 biospecimens from the current cohort. You can see the graph that shows the exponential growth. And the only way this could have been done is through collaboration, through the Cbtn mechanism, where the Cbtn institution sites submit their biospecimens and clinical data to the operations center here at CHOP. I have a small screenshot also from our website just depicting one of the projects and why Cbtn was important for that. And, you know, we get questions of, well, how many biospecimens do you need? And is 40,000 enough? And why do you need 40,000? And, you know, from this investigator, he was able to say that the Cbtn was the only biorepository with significantly large collection of these specimens for him to be able to perform a study. And that's the kind of feedback we love to hear. You know, we want to be able to empower the researchers' questions across the globe to really combat these diseases. You know, why do we collect saliva and spinal fluid and blood plasma from family members now? We do that because this is another data point for researchers. When researchers are looking at the mechanisms of disease, they need to also look at what's normal or what's normal for a family. And so comparing the tumor tissue and the genome of the tumor to the genome of the family provides the investigators another lens to understand what the tumor is doing and how it's evolving. One other resource that the Cbtn provides to researchers, in addition to biospecimens and this clinical data, are cell lines. And you may have heard them as preclinical models. And cell lines are extremely important to researchers because they essentially create a copy of the tumor outside of the patient. And so we have these cell lines within our lab, and we can grow them, we can generate data on them to understand again how the tumor is growing, and we can also rapidly test these cell lines and the tumors with drugs. So we can do very large drug screens where on clinical trials you may need to test one therapy against the other. In a cell line, we can test hundreds or thousands of therapeutics at one time. So cell lines are definitely a resource that wasn't available for pediatric brain tumors that Cbtn is trying very hard to be able to support the researchers across the board. I'm happy to report from those initial days in 2011 where we had maybe a handful of researchers across the US looking at brain tumors. We now have close to 200 approved scientific projects, and these are researchers from all over the world. It's fantastic to think that we have almost 200 projects, again, going on at the same time. Looking at that original model of one lab, maybe making 5 to 10 years to study something in a progress, we can say, through sharing in real time and collaboration, we have almost 200 investigators investigating these brain tumors, around the globe. We have a small chart here that shows the projects by the type of brain tumor. I also have a little screenshot here. If you go to our new website, you will be able to see and filter the projects that the Cbtn is supporting by brain tumor type, and whether they were specimen projects where we provided researchers specimens or we provided them data. So hopefully the website is a new resource to be able to understand the progress and the latest scientific projects that are being supported by Cbtn. Of those 190, I mentioned quickly the biospecimens. We've shared over 15,000 specimens to researchers to date. Again, you can take a look at the website to read about every single one of those projects and learn a little bit more. And just to be clear, these resources are available to any researcher around the world. They do not have to be part of the Cbtn member institutions that I mentioned earlier. Any investigator can submit a project and it's reviewed by our scientific committee. After approval, then we provide those specimens or data or materials free of charge to the researcher. What we do hope is that researcher will share back their information, their data, their learnings with the consortium so we can continue to grow together. At the bottom there, you see a couple of snapshots of publications from these projects. Again, they are available on the website. Specimens themselves are a depletable resource. We try very hard in the Cbtn to serve as many investigators that we can from one subject's biospecimen. And so that is a depletable resource. To take that challenge on, the Cbtn has really invested in foundational data. This data includes what we call whole genome data, where we sequence every single base pair of the genome of the tumor. This data can then be shared in real time with investigators, with as many investigators as request. The data itself is non-consumptive and can be used over and over. So to not deplete specimens and to make sure that we have reference materials available, we do create large-scale genomic and proteomic data sets to be shared around the world. We follow the NIH guidelines for sharing, which means if we provide raw genomic data, the investigator just has to attest that they can manage a raw genomic data. And we have 112 projects within that area. We also have what we call open access data. Now, this is data that is not raw, has been processed, and the NIH, the National Institutes of Health, which oversees all research in the United States, deems as able to be accessible just by registering on the website. And we're extremely happy to say we have about 5,000 researchers that have been to the website and have started to work on that data. We also have one other portal, the Kids First portal, with 1,500 registered users. And again, another area where researchers can come together online to look at the data available and be able to do collaborative data projects online in a shared research analytics workspace. Again, you can see the number of research projects, almost half are from non-member institutions. You can find all these projects online at our website, and as I'm showing here in the bottom right, you can also see the publications that have been resulted from these projects. We did get a question about some of our informatics portals, and one of our informatics portals is called Cbtn. Cbtn is a large big data platform for analytics, storage, compute, and sharing of data in process. When I started back in 2011, we actually had to download data to hard drive and ship them one at a time to researchers. And this process took more than 6 months. Now, with Cbtn and the portals that we have, researchers can log on, get access to the data immediately, and begin work. The question here was posed of, how can genomic data help us better understand pediatric cancer? I think I've touched on this a couple of times, but really it's understanding that mutation that the cancer has that our normal tissue does not. And how does that cancer use that variant and use that mutation to be able to overcome our normal regulation mechanisms within our cells and propagate forward. Cbtn works just not only with our member institutions, but we are happy to, and we are very excited to partner with the NIH, the National Institutes of Health, the governments in the EU, in Australia, with the leading data scientist collaboration groups, and then with patients and foundations and their organizations. You can see here a couple of the logos from the groups that we work with. And of course, we're hoping to scale and work with any investigator or group that is interested in collaborating and making sure pediatric brain tumors are on the forefront of the most innovative and cutting edge science. Our future plans are we have to keep increasing the number of institutions that are part of Cbtn, and this allows us again to collect those very precious biospecimens and that clinical data. We want to, of course, increase the number of projects. We're super excited to almost be at 200. I hope that we're at 10,000, or as many that's needed to make sure that we can tackle these diseases and find new therapeutics. We are working with new clinical trial consortiums, PNOC, Zero Childhood Cancer in Australia, and we are using the Cbtn data to help inform the development of new precision clinical trials, and also help with treatment decision support for clinicians on those clinical trials. We actually take the subject's genomic information and we compare it against the Cbtn data set. And by having those outcome information, we can provide the clinician with a more precise understanding of the tumor within that one patient. And as I said, we hope to continue to expand with our patient and foundation families, our governments, and we're looking at starting to work with pharmaceutical and commercial partnerships to expand those new discoveries. So I think at this point, I may be turning this over to the next speaker.
Yes, that would be me. Hi, everyone. Can you hear me okay?
Yep.
Okay. I am Elizabeth Everett. I have been with Cbtn since 2015. I'm just finding where I have slide deck. And I have primarily been involved in the research project. Sorry, I'm back to my slides. In the research project process, helping the investigators navigate our request forms and the communications with the scientific committee. And Jenna actually touched a lot on many of these things, but all of these resources that Cbtn and our partners have been generating, we obviously want everybody to use them because the more people that are looking at this, hopefully we will find cures for these kids faster. So these resources are available to any researcher, anywhere, anytime. We provide online portals to review what samples and what data we have that are available to access. Again, these are 100% free. Any sample processing that needs to be done before shipment, any shipments, that's all covered by Cbtn. We also empower researchers through our primary review process. Every sample request that we get goes through a primary review just to make sure that the investigators are using the samples to their best scientific merit, and researchers can contribute their expertise to reviewing these for their specific fields. And the data is open access and easy to use. We have monthly office hours where investigators can log in and ask any questions. We get them from how do I log into the portal to more specific questions about analyzing the data. We also have clinical trial working groups with the PNOC group, and if researchers are interested in specific brain tumor diagnosis, they can contact us to see how they can get involved. There's two ways to access the data for research. Jenna mentioned PC Bio portal, which is our open access data portal. All you need to be able to access that data is a Gmail account, I believe. So you can log in at any time and see that. For our other data, there is a form that we ask the investigators to fill out, and this is just to capture some basic information about what they're going to be doing with the data. We need a signed data use agreement, which again is just making sure that they can handle the raw data, and provided all of that is signed and ready to go, investigators get access to that data within 3 weeks. For the samples, we have a different portal. Currently, investigators can go to Harvest to see what samples that we have. They can create cohorts based on whatever diagnoses or other phenotypic data that they're interested in. And then it's the same form for samples or for data. That way, if investigators are interested in both, they're only filling out one form. And then the sample requests take a little bit longer for review since these are depletable resources. So it goes through, again, that primary review from someone who's an expert in that particular field. And then the rest of the Cbtn scientific committee will review the request, which takes about 2 months, that entire process. And then once approved, samples should be at the investigator's lab within 6 months, provided all agreements are signed. If anyone has any questions about that process, emails research@cbtn.org. And then we also have a link there if any institutions are interested in becoming a Cbtn member. For more information about all the projects that Jenna talked about earlier, those are all at our website. We also have more information about the samples and the data that's available, recent news, and how to partner with Cbtn. And I think I'm doing this slide as well. So we also are empowering patients and families. These are our most important partners in this research. Without them, we wouldn't be where we are today. The way we partner with them is through tissue donation and other biosamples. We do have ways for patients to donate their tissue even if they are not part of or not receiving treatment at a Cbtn institution. If you know someone who is interested, the email is operations@cbtn.org, and they can work with the patient and their clinicians to get tissue donated. Also providing the patient experience and insight to the clinical working groups, and then supporting the research projects that we have going on at Cbtn. And always advocacy efforts are important as well, because while this is a rare disease, it is not rare to anyone who is going through this right now. And I think that is it for me. Now it is open to any questions.
And Elizabeth, I think it's one thing I just wanted to add. My name is Jonathan. Jonathan Waller. I've been on the communications team for Cbtn for since 2018, and it's lovely to be here today. I did want to just call attention to one question that we have gotten from families about participating. A lot of families want to know, like, how do they contribute to the Cbtn's research if they want to donate biospecimens, genomic information, or even clinical data? And especially they want to know, like, how do they do that if they're not from a Cbtn member site? You know, not all children are able to go to Lurie Children's or CHOP or any of their other sites. However, though, you know, we do have staff at the operations center here that can reach out to patients' individual doctors at their home hospitals to consent over the phone. And then we'll be able to work with your clinicians to obtain any samples that are able to be taken during surgery, or to allow you to donate clinical data to our research studies. So again, you don't have to just be at a Cbtn institution to contribute really valuable resources to our efforts.
And Jonathan, I also received a question. I use the term, this is Jenna, I use the term proteomics, and I got a question to provide a little background on what that means, and thank you for submitting that question. Proteomics is the study of the protein, and the drugs are the drugs that we are used, they are targets for the proteins. And so we need to study the genomics and the mechanism that's going on to understand if they are creating a variant that then the protein that the drug would not connect to is there, or whether we need to establish another therapeutic mechanism because of the protein aberrations within a specific brain tumor histology. There has been a new publication recently released from the Cbtn. It was the first proteomic landscape on pediatric brain tumors. This study was able to identify that children looking at their proteome in addition to their genome did provide new therapeutic potential options for researchers and clinicians to further study. That publication is on our website, and I know that proteomics is going to be at the forefront of many projects to come within the Cbtn.
And so, you know, we do want to just kind of open this up to any questions from folks watching us today. Again, we want to thank you all so much for being here today. You know, this has been a really wonderful session. And yeah, so we just wanted to open up the floor and see if anyone had any follow-up questions for us.
I do. I'm Kristen. I'm going to ask what I just a follow up on what Jenna just described on the proteomics. So I guess as someone who knows very little about this, it kind of makes my head spin. So now I'm like, do they look at the proteins for the targets for the drugs? Do they look at the DNA, the biology of the tumor? Like, when you say all these things, Are they all different, or like the biology of the tumor? Does that mean the DNA and the protein? Like it's just that basic of an understanding. And like what drives the treatment the most? Is it the proteins, the DNA? Like, you know what I'm saying? If you
I do know what you're saying, and I guess I would kind of liken it to additional lenses. So the first lens a researcher may have is the genomic lens, and they can understand so much from that. And then they get additional layer of information of the proteome. That gives them maybe more information, more targeted information, or more information to understand what's going on. Then we give them maybe imaging data. So in the Cbtn, we collect imaging data as well. And so what they tend to do is then bring all of those data modalities together to actually understand the full complement of what's going on. I would say that you can't do one without the other. There are different areas of study, but bringing those areas of study together gives us a better understanding of that specific tumor. Is it a specific tumor that occurs in a specific location? You know, and because of that imaging, we need to look at what actually is going on in the genome. And then from that genome is the resulting proteins or lack thereof, or problems with the proteins. And the protein is where the drug actually connects to. So it's going to depend, Kristen, on these very complex tumors and what's the mechanism of action that they're using. And so, you know, bringing them together is what Cbtn is trying to do, provide all of this comprehensive data to give them a better picture, a clearer insight. And so I think, you know, some of us have probably heard that that explanation of, you know, we're in four people in a dark room and one has a hold of the elephant tail and another has a hold of the elephant trunk, and they think it's a very different animal, but you turn the light on and there we are together. So bringing those different modalities together would give them the full picture of what's really happening within the tumor.
Thanks.
And Kristen, you know, a really wonderful opportunity next week at 2 p.m. on the East Coast. We're going to be hosting a panel discussion with a number of the key investigators that were involved in a huge study that we did over the past year with Cbtn on proteomics. It's going to be hosted on our Facebook page. You know, again, it's in the afternoon, 2 to 3 p.m. However, it's going to be archived and recorded. So anybody who can't watch live, there's going to be an excellent discussion with these investigators, and they're really looking at, you know, what is the impact of proteomic research on childhood brain tumor tissue research, and how are those able to be used to bring us closer to better treatments. And so, you know, again, you know, if you want to, then our next slide, we kind of show each of the ways that you can connect with us and learn more. You know, you can follow us on social media. You can sign up for our newsletter. And again, you can continue to join continuous community information sessions. Our next one is going to be January 28th.
Hi, Jim Reamer, if I could ask a question, please.
Please.
I'd like you to compare existing treatments for other cancers, maybe in adults, you know, some other existing treatment where there's something that's out there, a drug is actually being used for a targeted therapy, and the process that was used to develop that, and the challenges that were presented for that therapy versus what you're trying to do now for children, and in the scope of the work that's required there. What I'm trying to do is get an understanding of, you know, how much are you up against? Is it just a matter of doing over something that was already been done for adults, or is this so very different in this in childhood brain tumors that the whole process is very different and the challenge is much greater? Thank you.
Sorry, I was on mute there. So I think that's a great question. And you know, there's no easy answer to that. So with the adult cancers, unlike the pediatric cancers, there's a larger volume of the adult cancers, right? So, you know, adult cancers, children do not get cancer because they're smoking or they're drinking or they're outside in the sun too much. The Cbtn leadership and investigators are really looking at this as a developmental biology disease. What's going on within the children as their bodies are developing that would attribute to this. There are very different mechanisms within the adult cancer that are also than the childhood cancer mechanisms. Now, we do lifespan studies, and we do across disease studies, because as we are looking down at the genomics and what we would say is the mutations within cancer, our opportunity in pediatrics is if we can find a mutation that is also found in adult cancer, whether it's brain tumors or not, there probably is an existing therapeutic drug available. And can we identify those mutations and then test those drugs in the laboratory on the pediatric brain tumors? To date, there was a variant found in skin cancer that was also found in pediatric brain tumors. Now, when this provided a lot of understanding of the mechanisms of that cancer in pediatric brain tumors, but it did not provide a therapeutic. The therapeutic actually accelerated the tumor growth in children, whereas it was you know, a good therapeutic in adults. So in adult cancers, you have a much larger population, and the origin of the cancer is different, has different mechanisms, can have different mechanisms. So for pediatric brain tumors, we have over 100 different types. And at any one hospital, you may see 10 of one specific type a year. So with the, you know, that's one of the challenges the Cbtn tried to overcome is, could there be a resource? So even if an investigator was interested in, let's say, studying, you know, a specific brain tumor type like medulloblastoma, how would they be able to get enough specimens? On the adult side, there's a population of patients, let's say in breast cancer, that's very large, and any one institution would then be able to collect the amount of specimens needed to do that research. So, you know, although this presents a great challenge in the pediatric brain tumor community, we're trying to look at that as an opportunity and really accelerate discoveries through collaboration. If Cbtn could create those resources and then provide those out to researchers where there has not been those resources before, could we then accelerate the number of projects that are happening across the board? So we partner with adults, but again, they don't face the challenges of the numbers, and there's different causes or origins for the cancers that are happening there. Here at the Children's Hospital of Philadelphia, we also do have a very large adult brain tumor repository, and we are looking at the lifespan for children to young adults, which the NCI, the National Cancer Institute, indicates goes up to 39 years of age, and then adults after that. And to try to look at the mechanisms that are happening across those different age variations.
Thank you.
The commercial partnerships have been slow in the pediatric landscape as well. Due to the volume of patients in adults, there has been a larger interest from the pharmaceutical companies to develop drugs and bring drugs to market. Within the pediatric cancer community, we now have new legislation called the RACE Act. And this RACE Act that it was put in place and really actually launched in August of 2022 requires pharmaceutical companies, if there is a variant in adults and they want to bring that drug to market, they now actually have to do the clinical trials in pediatrics to see if there's benefit for kids. In the past, they were able to receive a waiver because of the quote unquote rare designation of pediatric brain tumors. And other pediatric cancers, but now they will have to perform those trials. Cbtn is hoping to really be a support for pharmaceuticals to be able to complete those trials, as we've overcome the challenges they would need to do of collecting the biospecimens and data to be able to mine the data for targets and run virtual clinical trials on the data. So different areas, but you know, we're all in the fight together, and we're looking to share knowledge and information and ways to move forward the whole landscape.
Thank you. That's great. I didn't know that about that there was a RACE Act that did that. That's great.
Yeah, we are very excited about that to start initial discussions with the pharmaceutical companies and really make sure brain tumors are at the forefront of the RACE Act. Brain tumors, as you know, very difficult to study, very difficult to treat because of the blood-brain barrier. But hopefully the work that Cbtn has done will ensure that the pharmaceutical companies pay attention to brain tumors along with other pediatric cancers that may be a little bit easier to research or treat.
Any other questions in the last few minutes this evening?
I just have one more question since we're talking about the RACE Act, and it might be too specific. If you want to talk to me about it at another time, that's fine. I just saw Nancy Goodman, who was really the driver of the RACE Act, sent out an email that one of the companies somehow backed out of having to do the pediatric portion. Did you see that? And how is that possible?
So I did not see that email. We have been in talks with Nancy about quote unquote loopholes, and you know, our concern was making sure pediatric brain tumors, they didn't fulfill their indication, you know, in what I was indicating, maybe an easier cancer to investigate. And so we were working with her on that. I did not hear about this specific case, but you know, be happy to take a look at it, but I'm sure Nancy is going to be on top of it.
Yeah.
She's quite a force, and we're super excited to be partnering with her on that.
Okay, thanks.
All right, we really hope that this session was informative and helpful for you all. We want to make them better, so please send any feedback you know, to the group, and we will make sure we have appropriate topics or appropriate attendees that can present on the information that will empower you in the work that you're doing.
Okay. And again, Elizabeth, if you just want to like advance one more forward, if you want to reach out to us, you know, again, you can connect with us in a number of ways. You know, there is our social media where you can leave us comments. You can email the research team at research@cbtn.org, or you can email the administrative team, including the outreach team for these presentations, at operations@cbtn.org. If you'd like to follow along with latest news from cbtn.org, you can go to our website. At the bottom, you can sign up at our bottom of our homepage. You can sign up for our newsletter. And again, we are going to be doing these, more in the future, where our next community info session is scheduled for the end of January. And we hope you guys can all join us. And I think that's it for today. So we just kind of wanted to all thank you so much for taking the time this evening.
Thank you, guys.
Thank
This is
you.
very
Yeah.
valuable.
Thanks again.
Keep up the great work.